All right. I think we're going to go ahead and get started. So thank you all for coming. I'm pleased to welcome you all to this plant ecology double feature. So I'll just say both Genevieve and Sarah have been spectacular interns, and they have really great projects that you're going to hear about today. And we're going to lead off with Genevieve first. So Genevieve is, excuse me, Genevieve Triplett is originally from New Mexico and graduated from the University of Hawaii in 2023 with a bachelor's in botany. So as an undergraduate, she did quite a bit of research on amphistomy, which is this uncommon and very understudied plant trait where stomata are present on either side of the leaf. And she published this work this past year in American Journal of Botany. So when Genevieve arrived here at Archbold, she had trouble letting go of stomata and amphistomy and started checking out these various plant species to see if they had amphistomy. So in the early days of her internship, she would just go in the field with the crew and bring back a leaf from a species or two. And then a few hours later, she just nonchalantly walk into my office and announce that she made a new discovery of amphistomy. And this happened over and over and eventually became the project that you're going to hear about today. And as you're going to hear, there is a lot of amphistomy here in the scrub. So Genevieve has been wonderful to have in our program. She always brings a great attitude every day. Although be warned, she is a very sneaky, unsuspecting April Fool's prankster. She also has an amazing and uncanny ability to really capture plants. She's a really fabulous artist and has provided some of the new works around our lab. So after she leaves here, she'll spend a month in California with her former undergraduate advisor researching more stomata. And then she's going to start a master's program at University of South Carolina, where she swears she won't be working on amphistomy or stomata anymore. Maybe. And with that, I'll turn it over to Genevieve.
Is this on? Is it working? Okay. Yay. Do I have a laser pointer? Oh, that is not. Okay. 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 Hello, everybody. Thank you, Aaron, for that amazing introduction. And thank you, everybody, for coming to my intern presentation. I did it, as he so thoughtfully spoiled, on amphistomy. Is amphistomy an adaptation to fire? Hello. Okay. So the scrub is a very stressful place for plants to live. There's intense light, intense heat. There's nutrient deficiency because the sand can't hold the nutrients very well. And there's water limitation periods followed by inundation. And all that include, on top of all that, frequent fire. So one way that plants are able to interact with their environment and survive these challenges are through their stomata, which are small holes in a leaf surface that act as the interface of gas exchange. So through them, they're able to obtain the raw materials for photosynthesis, as well as anything to do with respiration and gas exchange. And then there's also transpiration that comes when any gases exit the leaf. There's always going to be some amount of water vapor exits it too. So what is amphistomy? Amphistomy is the presence of stomata on both surfaces of the leaf. Most plants you see out there have all of their stomata on the lower surface only. So this trait is surprisingly uncommon, and it's not super well understood. Oh, can't see me? Okay, sweet. And it's not super well understood why it's not always on both surfaces, because there's many benefits that come with amphistomy, including a shorter pathway of carbon dioxide to enter from both sides of the leaf to encounter these chloroplasts, which increases photosynthesis and may increase productivity. So that's clearly a benefit to the plant. So why don't all plants have amphistomy? It's because there's also costs, including increased water loss from the transpiration, as well as the upper surface being more exposed to the elements, such as sun exposure and pathogen exposure, as can be seen in these pictures where the upper surface has a lot more fungal filaments than the lower surface. And so this establishes a trade-off. Would plants rather have increased photosynthetic production or increased water conservation? And plants handle this trade-off very differently depending on the species. Everything from large sparse stomata to tiny densely packed stomata in this of hickory, and the others the saltbush, and pretty much everything in between. So there's two ways that I will be quantifying stomatal traits in this project. The stomatal ratio, which is the percent of upper surface stomata over all of them. So the higher the stomatal ratio, the more stomata there are on the upper surface. I will also be discussing the stomatal density, which is the number added between both surfaces divided by the area of the microscopic image. So this will give you the rote number of stomata per millimeter squared. And this can change in species in two main ways. There's adaptation, which occurs over a long period of time based on consistent environmental stressors. And then there's plasticity, where within one plant with the same genome, they're able to they're able to plastically change their stomatal characteristics just by differently transcribing their genome. So this plasticity is variation within a generation. And once a leaf is fully expanded, it can't change its anatomy. A epidermal cell cannot just turn into a stomata at will. And amphistomy is in general very understudied. We're not really sure what controls whether a plant has it or not and why it's so uncommon. And I've spent almost 4 years researching this question, from just doing what I was told in a lab to designing my own project to working on a project that is large scale across many different schools. So now that I have full control over my project, I wanted to know, could amphistomy be an adaptation to fire? So I will be looking at this two ways. One is the intraspecific variation in response to fire. So this will be looking at variation within a species, either population-wise or just in an individual. That is intraspecific variation. And you might be asking me, Genevieve, why would you even come up with this? Why would amphistomy be an adaptation to fire? Well, my thought was that the increased photosynthetic production that comes with amphistomy may give seedlings in a post-fire habitat a competitive advantage, because it gives more photosynthetic materials and allows for more rapid regrowth, or more rapid regrowth or more rapid growing from seed in general as a seedling. There are two species of palmetto here, and they exhibit this fascinating ability to be burned completely to the ground and sprout up like nothing ever happened. You've probably seen that burn that's just out there. The palmettos are already growing back. So they're really the first thing post-fire that comes. There's two species. There's Serenoa repens, which is the saw palmetto, and I'll illustrate it with that little diagram right there throughout this. And then there's Sable etonia, which is the scrub palmetto, which is that diagram right there. The scrub palmetto is much more narrowly occurring, and it, compared to the saw palmetto, it really only occurs in Central Florida, whereas the saw palmetto occurs in multiple states and all over Florida. So I was fascinated to compare such a narrow one to a more diverse one. But something in common with them is they both have underground storage in the form of carbohydrates that allows them to regrow quickly after the fire. And this ability for regrowth is not the only reason I decided to look at some stomata on palmettos, but also they act as a natural laboratory to study stomatal variation over time, because they keep their leaves for up to 3 years. And in those 3 years, they're constantly putting out new regrowth. And with each regrowth, they are plastically optimizing themselves to the environment they're facing at that exact time. So in looking at the leaves over the 3 years, you can see 3 years worth of leaf adaptation. here's a diagram of how they grow. As you can see, this laser pointer is not working. The newest leaf that's closest to the center is what I sampled as the pre-burn leaf. And then I burned it, and all the leaves are gone, except this one little compact leaf in the middle that has a burn scar, but it's still very much there. Now, I cannot say for sure whether this one has control over stomatal ratio and stomatal density, because since it's already preformed in there, the stomata might already be formed in there. And like I said, once it's fully expanded, it cannot change its anatomy. So what I did was I waited for that one to grow out, and it's easily distinguishable because of its burn scar. And then I sampled the next one that I'm 100% sure the leaf had control over. And this will be called the post-burn leaf. So throughout this presentation, I'll be referring to the post-burn leaf, and it's always the first fully formed one after a fire. And the way I got the stomata is by doing a stomatal peel on a consistent part of the leaf for every individual. And I used a nitrocellulose peel using clear nail polish and put that under a microscope and obtained something like this. As you can see, the two species have very different stomatal morphology, which is what inspired me to study both of them in the first place and compare them. Sable Etonia, the scrub palmetto, has much larger, less densely ones, and the saw palmetto, Serenoa repens, has more smaller ones, and these are the same scale. So to test this theory, I did two approaches. First was an experimental burn. So my goal for doing an experimental burn was to see, do palmettos have the same stomatal traits before and after they grow back? To test this, I sampled 6 palmettos. I burned them, waited 4 months for them to grow back for that second fully intact leaf, and I sampled the exact same plants in the same spot on the leaf. And I repeated this for a site where I cut all the leaves off instead of burning them to see if it was truly the fire causing this response or if it was just the act of regrowing. And here's the results. The burned palmetto's stomatal ratio, which again is the percent of stomata on the upper surface, stayed very consistent at around 50%. So 50% of the stomata were almost always on either surface. And this remained consistent before and after the burn, which indicated that a change in stomatal ratio was not necessary to survive this burn, but there was a little bit more variation afterwards. And for the control, largely the same thing is seen, as both bars are pretty much the same with no statistical difference. However, for the cut site, remember that burned leaf, I had to wait for it to grow out because it was already formed in there. The one analogous to the post-burn leaf, the second one, never grew back. So these grew back significantly slower. So it was the fire itself that was causing the rapid regrowth and not just removing all the leaves. However, I decided to include it in the comparison nevertheless, and before and after remains the same for stomatal ratio. The stomatal density is a different story entirely because both species experienced a marked decrease in a stomatal density after the fire, which means they had much fewer total number of stomata, which is likely because in this post-fire environment, they are prioritizing water conservation as opposed to having as many stomata as possible and prioritizing rapid carbon gain and regrowth. And this effect is seen in both palmettos, but is more significant in Sable Etonia, the scrub palmetto. And here it is for the control. The saw palmetto didn't respond at all to the control. The bars are the same heights. However, the scrub palmetto did actually respond to the environment, which means even in that little preformed bundle, it is able to control its stomatal density. And this is likely because it's not fully expanded yet, and it can just grow bigger to have less stomata per leaf area, which is why I bothered to include it anyway, even though it's not a comparable leaf. So in summary, the stomatal density decreased after fire, while the stomatal ratio remained roughly the same, but it did spark a little more variation. Additionally, the cut palmettos grew back much slower, and the two species reacted differently to the control, with sable Etonia reacting to the control and the saw palmetto not. So that's all fine and dandy, but I'm only here for 4 months to work on this project. And I want to see what the long-term impacts of this could be and the subsequent reacclimation after a fire. So for approach 2, I had to get a little bit creative and use a time since fire gradient. For this, I compared the post-burn leaf that I was able to access, and I compared it to the newest leaf. So the post-burn leaf is the oldest leaf, and it will represent the plant's immediate response to post-fire regrowth conditions. And looking at this will show you what it is prioritizing to survive losing all the leaves. The new leaf is the newest leaf after that, and it can be anything from the second leaf in ones that were very recently burned to a really like the 8th leaf or something. So it'll show you how much it has adapted within that time frame. And it'll show you how the plant has reacclimated, and now that it has more leaves, what is it prioritizing? So hypothesis 1 will be that the post-burn leaf will have more stomata, meaning it was prioritizing carbon gain, and now it no longer needs to after adapting. Hypothesis 2 will be that the post-burn leaf will have less stomata, meaning it may have been prioritizing water conservation, and now it no longer needs to. And hypothesis 3 will be that the old leaf will have the same amount of stomata, meaning it was either unaffected by the fire or the plant has not yet or will not reacclimate. So in order to test this, I used Archbold's 50 years of extensive fire records and chose 12 sites ranging from half a year to 50 years post-fire. And I compared the post-burn leaf to the reacclimated leaves. And here's what I found. This is the stomatal ratio, which again is the amount of amphistomy it has. And as you can see, there's a lot of initial variation and then a leveling out after the 3-year mark, where the post-burn leaf will no longer be alive, because remember, they only last 3 years. And this may look like a lot of variation, but I had to zoom in a lot to even see this. It's really only a 2% variation. So again, there's some initial variation, but it remains mostly consistent for both species. So in summary, the post-fire regrowth generates initial but minimal variation in stomatal ratio. And here is the stomatal density. This looks very similar, except the scale is much larger, and these are actual things happening to the leaf. As you can, especially in the scrub palmetto, sable Etonia, you can see a large difference between the old and new leaf within the first 3 years, which is not seen in the scrub palmetto. If you zoom into those first 3 years, you can see a trend where after 1 year, the oldest leaf has less stomata than the newest leaf. But then after 1.5 years or more, then the oldest leaf actually has more stomata than the newest leaf, which means sable Etonia has an initial decrease in stomatal density followed by an increase. And Serenoa repens, the saw palmetto, has its regrowth is more variable, but there's really no trends to be seen here. So for part 1, the intraspecific variation, the summary is that sable Etonia has a lower stomatal density post-fire, suggesting it's prioritizing water conservation rather than increased carbon emission. And Serenoa repens has a less pronounced reaction overall. Additionally, a significant change in stomala ratio does not appear to be necessary for post-fire recovery. And by the time the post-burn leaf dies, the traits have generally restabilized. So that's so cool. But the format was messed up. But I cannot possibly say that amphistomy is an adaptation to fire just by looking at one species. So my next step was to look at how common it is in the scrub entirely. So my approach for this was to get 100 species of plants and deduce whether they have amphistomy or not. In reality, I hit 117, which happens to be how old Richard Archbold would have been this year. Happy birthday. And as a reminder, amphistomy is not super common. It's really hard to find a worldwide estimate, but this paper from 1927 says maybe a quarter of British angiosperms have it. And some personal communication I had with researchers looking at grasslands in China said up to 50%. And then another one estimated about 10% worldwide. But our species here, 62.9% of them have amphistomy. That is so crazy. It's like remarkable. Like, I was always told that this was a really rare thing for species to have, and here we are with almost 2/3 having amphistomy. So then I decided to, wow, you can't read that at all. I decided to pare down a phylogenetic tree from Zan et al. And kind of map this trait on the phylogenetic tree to see if there is a signal detected. And as I found, there is a very strong signal detected with a p-value of 0.001, which means this is an evolutionary thing where families kind of have it across them. So it's not a hard and fast rule. And it didn't just evolve once. As you can see, there is a lot of groups of amphistomy and no amphistomy all the way up, which means the trait is either re-emerging or it is being turned off and on genetically. The second option is most likely, as previous work I have done has been working on genes that are able to turn it off and on, but it's incredible that it's even working at a cross-species scale. And the family that had the most amphistomy was the bean family, Fabaceae, and the one with the least was the oak family. So then I decided to look at amphistomy as an adaptation to fire. And so first I had to look at whether plants that were significant or actually adapted to Florida had it or not. And I found that no matter whether it was endemic, native, exotic, or invasive, there was still the majority of species had amphistomy, which implies that no matter where these species are coming from, it is a helpful trait to have in the scrub environment and to be able to live there. And then I found that plants that reseed after the fire, 81.8% of them had amphistomy. That is the vast majority. And as a reminder, I looked at this in the first place because I thought amphistomy might be helpful in a post-fire habitat with plants that reseed themselves and have to grow and compete after the fire. So that is a point towards that guess. So in summary, amphistomy is a surprisingly common trait in the Florida scrub, and it appears to be beneficial for plants that die and reseed after a fire. And in conclusion, amphistomy is likely an adaptation to the challenging conditions of the scrub, which includes fire stress. Just using this alone, I can't say for sure whether it is the fire. Further work must be done, but this is a great first step in determining whether fire is the main cause of amphistomy. I would like to acknowledge that this work took place on indigenous lands, especially of the Seminole and Miccosukee tribes. And acknowledgements. Thank you to everybody.
Thanks so much, Genevieve. It's awesome. I'll go around with the questions.
Hi. Can you describe why, like, the percentage for the reseeding for the, I can't say the A word, does that in the reseeding species is much higher than for like the resprouting species?
Um, um, all of this is conjecture because I can't say for sure, but the reseeding species have to go from just a seed to a whole plant in order to compete, while the resprouting species, they are already there and they're already established and they just have to grow new branches. So they don't have to prioritize in growing back as fast. So they often are the ones that regrow first, as in with the palmettos.
We'll do that. See you then.
Fascinating. I loved it. Why was the density seemingly more responsive than the ratio?
Oh, I forgot to mention that. I think it's because the ratio is kind of like a set trait. Uh, none of the research I've ever seen has shown like a change in the model ratio within an individual. So this is kind of finding that it's a set trait, but the density is really what's responding. And even though the stomata are decreasing, the ratio staying the same means it's decreasing on both surfaces at the same time rather than this one surface losing it or not.
Great job.
Yay!
I'm wondering if you know if there's a trade-off between amphistomy and stomatal conductance. So like, let's say there's all of a sudden much more stomata. Do they then have a lower stomatal conductance, or does that remain the same?
What's that?
Oh, stomatal conductance is like the aperture or the opening of the stomata, and so that will impact how much gas exchange is able to take place.
Yeah, so previous work I've done has found that amphistomy does increase conductance and photosynthetic rate. But I'm not exactly sure if like more amphistomy will cause individual stomata to conduct less. That's a good question.
Hi, Genevieve. Yeah, really cool talk, really fun results. I was wondering if there's anything else that sort of correlates with stomatal ratios like leaf thickness or leaf size or anything like that. Yeah.
Thank you for asking. Come on.
I promise this wasn't planted.
No. Why won't I'll do it here. So I actually took this out of, why isn't it working?
Oh yeah.
Oh,
Oh, sorry. Wrong end. Oops. This one. You can just.
okay. Well, I took it out of my presentation for time's sake, but I found that plants that have all of their fuzziness, like it's called pubescence when a leaf is fuzzy, ones that have all of it on the lower surface were 100% of the time hypostomatous, which means stomata only on the lower surface. And ones that had fuzz on both surfaces were almost always amphistomous. So the fuzz is really on the side that the stomata are, which makes sense because fuzz is used to decrease transportational water loss. Yeah, and I also found that woody species, this is a trend that's always been marked. Woody species almost never have amphistomy, and herbaceous ones often do. However, I found that even a quarter of the species here that were woody had amphistomy, which means the like pretty much 0% of the time is actually a quarter of the time here, which still implies it's useful here that even that many have it.
Oh, there it is.
Oh.
No. That's my Genevieve's one.